Hi, everyone. Welcome to the population genetics session at Virtual Evolution. We have 5 talks, and our first speaker is Kavita Jain. If you want to go ahead and share your slides and start.
You confirm it's shared? Okay. Yeah. Okay. So good morning, good afternoon, good evening. I'm Kavita Jain, and I will speak about what is the effect of changing environment on site frequency spectrum. And this work is done with Sachin Kaushik, who is currently a postdoc at Cornell. Okay. So as all of you know, that there is a lot of genetic diversity in natural populations. So, a lot of genetic diversity in natural populations. So here is some old data, which on Drosophila species, and where you can see that many of the species have a very high diversity, sometimes as high as 3% per site. So of course, one would like to understand what evolutionary forces shape this diversity in a population. So of course, the mutations, they are the ones that create new variations, they create diversity, but this diversity can be lost because of genetic drift and also because of various forms of selection. Now, so therefore, there is a force which creates diversity and the forces that reduce or lose diversity, and it's possible that a balance may be reached between these two forces. And in that case, one can speak about or think about equilibrium diversity. However, it is important to understand that many times populations may not be in equilibrium. So that could be simply because they're in some kind of transient phase and the population is approaching, it's equilibrating towards this, this stationary state, or it's possible also that the population may never reach equilibrium. So one should distinguish and remember that there is 2 types of situations, equilibrium and non-equilibrium. Non-equilibrium is the ones in which the quantities are time dependent, and I will speak about both the cases. So now there are various measures of diversity, and I will focus on one, which is the site frequency spectrum. And just to be clear about what I'm talking about, here's a little example. So I have just, let's say, 6 samples, and let's say there are only 8 polymorphic sites. And site frequency spectrum simply means counting the number of loci that have I number of mutants. So assuming that 1 is the mutant and 0 is the ancestral allele, here we see in this example that there are 4 loci that have 1 mutant. There is only 1 locus which has 5 mutants. So that's here and here, and then one can basically use the data and find out the site frequency spectrum. So that's from the data, and one would like to understand what is the shape of this and what evolutionary forces are acting to give a certain shape. Now, let's start with the simplest situation in which let's assume that there is no selection, that is this, and also make other simplifying assumptions such as there's random mating, diploid, parametric population. Furthermore, I'll assume that the genome is modeled by the so-called infinite sites model. What that means is that the sites are unlinked, there's no linkage, and also because the number of sites is infinitely large, so whenever a mutation comes, it occurs at a new site. Okay, so this is the usual classical infinite sites model. And it turns out that in this case, one can show that indeed SFS reaches an equilibrium state. And this is the characteristic shape here shown in blue, and it's a monotonically decreasing function, which shows that at most sites, mutants remain at low frequency, and that makes sense because, because of genetic drift, they cannot reach high frequencies. So this is the characteristic, very well-known result. And of course, SFS is distorted by various factors. For example, due to high mutation rates. When mutation rates are high, then the infinite sites model assumptions no longer hold, and there could be corrections due to that. Very important factor is, of course, when there is linked selection. And there's a whole lot of literature which shows how SFS is distorted due to those reasons. But today's talk, I'm interested in understanding how changing environment affects the SFS. And since we're assuming neutral case, what is relevant here is to understand how demography might affect the SFS. In general, when there is demography, SFS will not be in equilibrium. Only under certain choices of N of t, it is possible to have an equilibrium SFS. So if the population size is fluctuating in time, then it turns out this, if you do have an equilibrium SFS, or suppose the N of t is changing periodically, one can think of some kind of time average quantities. So these things have been worked out, and what turns out is that the answer is same as that of equilibrium SFS, the one that I showed you just now, just before. The only difference, however, is that there's an overall reduction because the census population size is replaced by effective population size, which is simply the harmonic mean of the changing population size. So this is a classical result, which is known for a very long time. Let's now bring in selection into the picture, and I'm particularly thinking of directional selection. If there's positive selection, then in that case, the mutants can also reach high frequencies, and that results in a U-shaped SFS. Whereas if the selection is strong and negative, then just like in the neutral case, the SFS is only decreases monotonically. Also, it decreases much faster than the neutral case. Only in the last few years, only people have started to think about changing environment in conjunction with selection. So there is some work in which demography is taken care of, but selection is constant, and similarly fluctuating selection, but the population size is kept constant. So then there's a natural question to ask, what happens when we have changing selection as well as demographic? So this is a model, that simple model that we consider. As before, we consider infinite sites model, that is again no linkage and mutation is at a new site. And at every site, we run a Moran process in continuous time, where the mutant homozygote has a fitness 1 plus S of t. The heterozygote has a fitness 1 plus H times S of t, H being the dominance coefficient. And in this model, not only the selection coefficient, but also the population size are time dependent. So one can run this simulation and keep track of polymorphic sites to find out the time dependent SFS as a function of frequency and as well as the time. What we actually did was, because we were interested in getting analytical results, we studied this problem in the framework of diffusion theory. So what has been shown is that the non-equilibrium SFS obeys this diffusion equation. So this is a sort of a standard equation in the subject, in which one has this, the SFS, which I denote by F. It has these two terms on the right-hand side. The first term is simply the selection term, and the second term is the usual drift term. The difference from the usual cases is that now selection and population size are time dependent. So this is about selection and drift, but because of polymorphism we also need to model mutations. So we use this idea of Evans et al., who modeled the mutational input through a condition at low frequencies, which is given by this expression, which is simply basically the equilibrium SFS in the neutral case. So we analyzed this model, but of course we cannot solve this equation exactly. So we made some choices. We assume that both selection and population size are changing periodically in time. So this could happen because of seasonal variations, for example, for the selection, and demography is also periodically changing, as has been seen, for example, in certain pre-predatory dynamics. So in other words, we assume that S and N are changing our sinusoidal functions, which are oscillating about certain average values. This model has quite a few parameters. First of all, time scales, they're the usual time scales, namely the drift and selection. Not only that, we have two additional environmental time scales because of these frequencies at which the selection and the population size is changing. It turns out it also matters whether the selection remains always positive or always negative, or does it change over a period? And of course, when it changes, sometimes it's positive, sometimes negative. That's an interesting situation. Selection and population size can also have phase differences. So there are several parameters and several scenarios and so on, but I will focus on a very tiny part of it and just tell you one or two simple things. So I'll just talk about the case when the environment is changing slowly. So what does that mean? So I'm thinking about, I'm counting the time in units of generation. If environment is changing slowly, what it means is that in a year, there are many generations of the organism that you may be thinking about, so that from generation to generation, selection is changing only a little. So in that sense, the environment is changing slowly. We can show that in this case, the non-equilibrium SFS at time t is well approximated by the equilibrium SFS with the instantaneous value of the selection and population size. And that makes intuitive sense because, you know, things are changing slowly. So therefore, the population can see only the instantaneous values. Okay, so that's the non-equilibrium part. But to get some more understanding, one would like to work with quantities which are independent of time. So to understand that, another point that we notice is that if selection is strong, then the deleterious part of the selection cycle, if the selection is changing with time, in sign, then deleterious part of the selection cycle does not contribute to polymorphism. Again, that's something intuitive and you expect, right? So this simple observation, however, has an immediate consequence, which is the following. Suppose you have a selection which is on an average neutral. So half of the time is spent in the positive cycle and other half in the negative cycle. And of course, we also have the demographic. Then if we look at the time average SFS, although the environment is neutral on an average, the time average SFS is not monotonically decreasing. Rather, it is a U-shaped function, as we expect for a constant positive selection case. Okay, so that's the first observation. Not only that, there's a quantitative difference. The answer for the changing environment, there's an overall reduction in the SFS compared to the case when the environment is in the constant positive selection, and that is captured by a different new effective population size, which is given by the arithmetic mean of the population size calculated in the part of the cycle where the selection is positive. So that little expression one can write down explicitly is difficult to calculate, but it's possible to make some to get some understanding. Okay, so this is the sort of part when the selection and population size were changing slowly. One can also go to the other extreme in which the environment is changing very rapidly. When environment is changing very rapidly, then population simply sees the average environment. In that case, the SFS would be the same as though the population is static and you just see in the mean environment. However, if you look at intermediate time scales where it's not too slow, not too rapid, then again we find SFS is U-shaped pattern. Okay, so these are the simple couple of things that I wanted to say. So all I wanted to say here is that if you have a U-shaped SFS, it's consistent with constant positive selection. Also, as in earlier work, and also our work, it shows that this U-shaped SFS is also consistent with varying selection. And as far as I know, there are no tests to distinguish between these scenarios, and of course, that will be very important and useful to have. The demography, however, unlike selection, changing selection, which has a qualitative effect on SFS, demography has a rather more subtle quantitative effect. For weak selection, demography effect is captured by an effective side, which is given by harmonic mean. For strong selection, it is given by an arithmetic mean. We can show this when the environment is changing slowly, but for the mathematically inclined, let me just mention that when we have intermediate timescales, we don't have analytical expressions, and that's again something which will be worth having. Okay, so that's the first line, that namely the varying strong selection affects SFS qualitatively, whereas demography has a quantitative effect on it. So this is just one line summary that I would like you to take home. With that, I will stop here. Thank you very much.
Thank you, Kavita. I think we have time for a question, and
So I see Joanna had raised her hand.
Yes, Joanna has.
Okay, so if I understood correctly, the only thing maintaining polymorphism here is recurrent mutation. So I was wondering whether you've tried putting in cumulative overdominance or reversal of dominance, or or a temporal storage effect from ecology, or something that would lead, I mean, my intuition is that those sorts of things might change the SFS, and a kind of characteristic of this, in sync fluctuation of demography and selection.
Yeah, so I guess you're thinking of balancing selection. So what I have done here is only directional selection. And the effects that you're talking about, we have not thought about it at all, but I guess it is worth thinking about, but I haven't thought about it at all. Yeah.
Okay, great. Any more questions? Okay, in that case, I think if you want to stop sharing and we can move on. So our next speaker is Joanna Maisel, who just gave the nice question. Let's see.
Thank you. So I have a bit of a mouthful of a title here to make it a little bit more accessible. What I'm really talking about is what happens when mutation rates are really high, super high, as in, like humans, sort of realistically high. And estimates, the most cited estimate we have is that on top of however much genetic load you get from both of your parents, there's on average at least, an average of at least 2 new harmful mutations. And if you delve into that literature that estimates that rate, genome-wide deleterious rate of 2, it actually makes a lot of conservative assumptions. And so some people like Alexei Kondrashov has argued that that actually a value of 10 is more realistic. But in any case, a genome-wide deleterious rate of 2 seems to be a lower bound estimate for humans, and we're not the only species like that. The problem is that, as again, Alexei Kondrashov has pointed out, evolutionary theory really has no theory that can cope with this high of a deleterious mutation rate. If you run simulations of this, they will just keep declining in fitness. That basically one intuition of this is it takes on average 1 selective death to clear 1 deleterious mutation. And, you don't have more selective deaths than you have population size. You just can't do it for a value greater than 1 and the population will decline. So the various possible answers have been posed to the question, why are we still here? So some people said, oh, well, I'll just normalize my simulations. I'll just have declining relative fitness, but it doesn't care. Fitness is divided by the mean keeps going down. I'll just normalize it, which is fine if you're doing a simulation, but really, that's not, as a model of reality, that doesn't make a whole lot of sense. When it's continuously increasing, you can say it's responding to an environment, but when it's decreasing, the environment's constantly improving and we have to evolve less, doesn't make a lot of sense. The dominant view is probably that it's synergistic epistasis that deals with it, and the papers that found this used extremely strong forms of synergistic epistasis, like everyone above a certain number of mutations is dead and everyone below a certain number is fine. The problem with this is we since found out that if you plot the fitness against the number of mutations, you get something terribly, terribly close to non-epistatic, to simple multiplicative effect. And what we've observed is just nothing like what those models seem to require. So digging in a bit more, what sort of mutations is it that are accumulating? So here, if you take the selection coefficient on the x-axis and you take their impact on the y-axis, and their impact is how likely are they to fix and what is their effect, just with Kimura's equation, what you see is that selection, if it can't purge everything, it's probably purging the strongly deleterious. And there's some sweet spot of slightly, or not so sweet spot, of slightly deleterious mutations that, that aren't properly purged and that do accumulate. As opposed to the beneficials, which just, the more beneficial they are, the more likely they are to have an impact. So our answer to this question of what stops these small deleterious mutations from accumulating is that nothing does. Let's just assume they do accumulate, and that's an ongoing process whenever the genome-wide deleterious rate is more than 1. And instead, the idea is that the beneficials, as you'd expect from Kimura's equation, are not remotely in any kind of detailed balance. There's no sort of pattern of deleterious fixation followed by exact reversal within a finite sites model. Instead, you have completely different compensation for many mutations at once. And so, you don't need as many beneficial fixations in order to just keep up with the fitness problem. This has been proposed before, for example, by Mike Whitlock, but his treatment of it mathematically was very generous to adaptation. It was of independent sites, and we know that clonal interference severely limits adaptation, and so we wanted to simulate that. And that meant seriously upping our game in how simulations are done. So if you, take your standard program like SLIM or something that to do forward time simulation, it cannot handle a deleterious genome-wide, a whole genome with this sort of deleterious mutation. It's just too many segregating sites to be tracked. So we built our code where we consider the genome to be a set of linkage blocks, and there's recombination at hotspots that are in between each linkage block. And that means we don't have to track every single mutation separately. We can just sum all of their mutational effects within each asexual linkage block. And conveniently, there's a bunch of theory that suggests that chromosomes actually do behave something like this way. Our code is integrated with tskit and all the cool stuff from tree sequence recording. And so we simulate, this conservative rate of deleterious mutation, a human-like DFE, and we don't really know what the beneficial parameters are, so we consider a range. And so this figure I showed you before is actually from our simulations. The mean effect of deleterious mutations is sort of off the chart here, and it's these slightly deleterious that fix, and the beneficials that fix are also much greater than the mean. And you see this extremely pronounced asymmetry going on in the simulation. And importantly, this is sufficient for us not to go extinct. So everything below the black line has constantly deteriorating fitness. Everything above the black line has constantly improving fitness, able to counteract evolutionary environmental change. And so taking a range, the red dashes are sort of our guesses a priori on effect sizes. The beneficial mutation rates is less clear what they should be, but these are probably on the lower end of what a genome-wide beneficial mutation rate is. And the bottom line is we seem to be fine. At least we can stay afloat. If you take, what quantitatively is our rate of improving, you can see the difference, between how long it takes us to get 10% better with and without our load of this conservative load of deleterious mutations. And you can see that we're going at least 13% slower than we would if we weren't subject to all these deleterious mutations on this lower bound. So now let's talk about some of the implications for this. So the way I see this is a form of complexity ratchet to have this asymmetry that, you can see lots of deleterious as inefficient introns, and then fixing the spliceosome fixes many at once, or lots of inefficient protein folding and improving the chaperone improves many of them at once. So, chances are that the beneficials are happening at a different level of organization to the deleterious, at least to some degree, and that could lead to an increase in molecular complexity. And importantly, unlike Mike Lynch's drift barrier theory, population size, neither census nor effective population size, does not play a critical role in this complexity ratchet. So the driver of the complexity ratchet is that the deleterious mutation rate is above 1. There is some role for NE and N, both of them, but they're on opposite sides of these two forces, and they're not critical, and that this complexity ratchet can happen actually at any population size at all. So, in contrast, Lynch's drift barrier theory puts a really central focus on census and effective population size and the link between them. And there the idea is that effective population size increases the genome size, which automatically entails an increase in complexity. And that complexity constrains the range of life histories that are available. For example, increasing body size such that census population has to go down. And so you get this positive feedback loop that way. And then as a byproduct, Lynch stresses that low effective population size relaxes selection on the point mutation rate, which can rise to much higher levels because selection isn't as good as keeping it down. I'd like to point out that's not the only relationship between these two variables. The causality actually goes quite strongly in the other way. When the deleterious mutation rate is above 1, the force of background selection is really strong because selection can't purge everything at the same time. So definitionally, the effective population size goes down a lot when UD is large. And then you again get that increase in genome size from not purging small insertions, but that increase in target size, irrespective of point mutation rate, itself increases the genome-wide deleterious mutation rate by increasing the size of the genome and the number of targets that could have a deleterious gain of function. And so what you get here then is there's two ratchets here in our view. There's a mutational ratchet that provides this high deleterious mutation rate, and then the complexity ratchet causes ratcheting, levels of complexity at molecular level on account of that high deleterious mutation rate. And the contrast here to the drift barrier theory view is that we really sidelined the role of ecology and the role of census population size, that those are not required in order to get this evolution of ratchetingly high levels of molecular complexity. It can happen for any population size. As a side note, fitness actually directly causes changes in population sizes. And there's just for fun, the causal diagram I have here also includes mutational meltdown, coincidentally also studied by Lynch. And in person, if you're in Albuquerque, my postdoc Walid has a talk on that. So to sum up, we don't think anything stops small effects that are deleterious from accumulating. We think they do, in fact, accumulate. And that they are compensated for and not reversed. And that is the driver of a ratchet of increasing molecular and cellular complexity. The high UD, which is the only condition required for this to happen, that can come from another ratchet or positive feedback loop involving the target size and background selection. And yeah, this collectively offers an alternative to drift barrier theory to explain pretty much the same observations. If using our simulation code sounds like a good idea to you, we think that it's really good that I think that the backdrop of extraordinarily high deleterious mutation rates is something that is a critical factor behind a lot of phenomena that we care about as evolutionary biologists. I personally was partly interested in sexual selection and good genes hypotheses. That was sort of one of my original motivations for doing this. But there are far too many questions, I think, that depend on the genetic variation caused by high UD. I can't possibly study them all. I won't be in Albuquerque, but as I said, do talk to Walid, who is there, and just shoot me an email, and I'm happy to meet new people over Zoom if anyone wants to, give our code a spin. And I'll just say this work was pretty much all done by Joseph Matheson. This really should be his talk, but his baby is actually showing up on time today, which is great. And so obviously his paternal leave has started and I'm giving the talk instead. So thank you all for your attention.
Thanks, Joanna. So we have a couple minutes for questions. If you want, you can use the raise hand button at the bottom. I can ask a sort of, we have one from Jeff. Let's see, ask to unmute. There we go.
Hi,
Right.
thanks. Thanks for the great talk, Joanna. You had mentioned clonal interference. Is that like frequency dependent selection? And do you, with your simulation, account for frequency dependent selection, or is it all direction?
So clonal interference is not frequency. First question, it's not frequency dependent selection. Clonal interference is just the idea that if A is favored and growing in frequency and you've got some exponential growth of that, and B is also favored and you've got some exponential growth of that, without sufficient recombination, the numbers that have both A and B is going to be smaller than you'd expect from independence. Recombination is in effect, it doesn't keep up with the rate of new beneficial mutations. And so the total fixation of beneficial mutations is lower than you'd expect if they were each operating independently. So it's the non-independent evolution of sites. If you can think of it, it's about linkage disequilibrium. They're positive, helpful for adaptation, linkage disequilibria, and unhelpful ones. And the helpful ones all finish and the unhelpful ones get stuck. And at any given point, there's a huge accumulation of unhelpful linkage disequilibrium in any given population because recombination is not infinite. So as for frequency-dependent selection, you can add that to this list of topics that one could look at. I am interested in particularly in a completely separate research program. I am interested in various things that maintain diversity in populations, in particular with temporal variation. But no, there's none of that.
Joanna, can you explain briefly why Slim can't handle this and what the leap is that lets the new simulation code do so?
Yeah, so it's basically Slim scale doesn't scale very well with the number of non-neutral sites to be tracked. And what our code does is it doesn't track them. It's not that, it uses linkage block approximations in order to avoid tracking every single deleterious mutation.
All right, thank you. Let's see. Our next speaker is Emmanuel Meram-Veliotakis. So if you want to share your slides, Emmanuel. Oh, I have to do this again.
Oh, can you see my, I cannot see my, okay, sorry. So can you see my presentation?
Yep.
Perfect. So hello everyone, I'm Manolis Meram-Veliotakis, and I will talk to you about how habitat persistence can affect population dynamics. So first, a few words about the habitat persistence hypothesis. When we are thinking about habitat persistence, we are thinking about stable versus unstable habitats. And an unstable habitat is one that is prone to disappear or undergo frequent catastrophic events in an evolutionary timescale, while a stable one persists for long periods of time due to its high spatial and temporal continuity. So a core expectation of this hypothesis is that taxa that specialize in unstable habitats should have higher dispersal propensity in order to escape these deteriorating conditions, while this is not true for the stable habitats. And if you imagine an island, an example of an unstable habitat would be the coastal sand dunes, which go through different succession stages until they disappear or undergo frequent catastrophic events from natural forces, like, for example, the sea level changes, while a stable habitat would be any inner island, more protected habitat types like the shrublands or forests. And the good thing with islands is that if you are working in an archipelago, you get natural replicates of your system in order to test your hypothesis. And this is what we did. We are working in the Aegean Islands, which are located between Greece and Turkey in the Eastern Mediterranean region. And there we sampled these tiny beetles that are ideal to test the expectations of the habitat persistence hypothesis, as they have one clade that specializes in the unstable sand dunes and one that specializes in the more stable inner island habitats. And apart from this difference in habitat preference, they are equivalent in terms of dispersal ability. They are flightless, they cannot really disperse that much, and they are also almost identical in morphology, and they occupy the same trophic and climatic niche. However, the most interesting part of this system is that if we look at their phylogeographic pattern, we see a very significant contrast. And for example, in this CO1 gene tree, if you see at the unstable habitat, the unstable clade has a very shallow genealogy that also does not correspond to the paleogeography of the Aegean Islands, while in the stable habitat, you see many different lineages, these green clusters, deeply divided, and also they occupy small groups of adjacent islands, each one of them. So to the extent that this can be representative for the speciation process, this difference could be because of differences in the success of speciation between the two habitat types. And from what we know, the success of speciation can be significantly affected by population isolation and persistence. So if habitat persistence could affect these two processes, it could potentially have an effect on the success of speciation itself. So we started this work with different expectations for the two habitat types. We expect stronger isolation and higher persistence for the local populations in the stable habitat, since populations can persist without the need to disperse, and also any possible catastrophic event is not expected to be of high frequency. While in the unstable habitat, we expect weaker isolation due to higher dispersal propensity, and also lower persistence of local populations due to frequent extinction and recolonization events. And these events, we expect to distinguish them as bottleneck events in the demography of those populations. So to see if our expectations were met, we sampled 47 populations across 14 islands in the Aegean. You can see the different populations with either yellow shapes, which is for the stable habitat, or blue shapes for the unstable sand dunes. And you can also see with green and blue shades the possible connections of the islands based on their bathymetry due to changes in the sea level. So the details are not really important, but the important part is that we have three island groups based on, let's say, isolation intensity. So we have the more isolated western islands, which, as you can see, have no connection with other islands regardless the sea level. We have the central islands, which are the less isolated, as they have spent more time being connected rather than isolated, and we expect them to behave as a single unit, a single island. And also we have an intermediate case, the northern island group, which have dynamic barriers among them and also between them and the central islands. So from these islands, we collected 318 specimens, more or less divided in half between the two habitat types, and we generated genome-wide SNPs using DDRAD sequencing. And we use these neutral SNPs for data analysis to test our ideas. So starting with population isolation, the first thing that we looked at was population divergence. Here you can see two heat maps using the same color scale. So with lighter colors, you can see in the unstable habitat lower values of DXY, lower population divergence, while in the stable habitat, you see in general, higher divergent and also a hierarchical pattern. You see that the most divergent are the Western islands, which is expected as they are the most isolated ones. You can see intermediate values for many between island comparisons, and the lowest values, you can see them in these edges, which are inner island comparisons, and also the central islands, which as I said, we expect it to behave as a single unit. And a similar hierarchical pattern is we can see if we look at the population structure across these islands. For the stable habitat, again, the Western islands are a cluster on their own, and everything else is a second different cluster. And if we infer the structure within the second cluster, we again see strong structure across the population, which corresponds to either single islands or adjacent group of islands. While this pattern is not present in the unstable habitat, for which we see no relevance to the paleogeography of the area, and we can see three different clusters, and depending on the population, different admixture proportion. So both of these results point out to the fact that it seems to be higher migration indeed in the unstable habitat, higher gene flow, and to explicitly test this idea, we created two population demographic models using FASEM-COAL, and we created 4 different models with different migration rates through time. And we have two simple models that favor isolation, one with complete isolation of the diverging populations, and one with a small period of migration with yellow and contemporary isolation, and also two models that favor dispersal, which is either constant migration or contemporary migration after a period of isolation. And what we did was we selected a population with at least 7 individuals. We calculated the joint site frequency spectrum, and from all the combinations, we selected the pairs with at least 2,000 SNPs per population within the pair. And this provided us with 101 different combinations of our populations. And for those, we performed parameter estimation and likelihood maximization using coalescence simulations with FastSimCoal. Then we calculated the AIC, and we proceeded to model selection based on these values. So, sorry. So here you see the results of this model selection in the form of a heat map. Each column represents a pair of populations, a combination, and what you should look for is the black tiles. These tiles correspond to a delta AIC value of 0, and in other words, this is the model that is selected for this pair. So you can see that in the unstable habitat, we have a pattern of contemporary migration being selected, while in the stable habitat, we see mostly isolation when we look in between island combinations, while constant migration mostly for within island comparisons. And so the takeaway message from this analysis is that it meet our expectations of stronger isolation in the stable habitat, and the barriers seem to play an important role for stopping gene flow between populations, or at least having some effect depending on their intensity. While in the unstable habitat, regardless of any barrier to dispersal, we see that gene flow keeps happening between the populations, and the isolation is either non-existent or very weak. So with that, we can move on to population persistence. And the first thing we looked at is genetic diversity, which can be an indication for that. So what you see here is each bar represents a local population, and with yellow, you can see local populations in the stable habitats, while in blue, you can see the unstable habitat populations. And again, we have a pattern here that we consistently estimate lower local genetic diversity in the unstable coastal sand dunes. So in a similar modeling approach as before, this time we created two population demographic models, but we looked at fluctuations in the population, effective population size. And again, we have two high, what we consider high persistent models, which is either one of constant effective population size or events of population expansion for our two populations, and two low persistence models that is one of population contraction or population bottleneck. And again, we followed the same model selection procedure, and what we see is the bottleneck model of low persistence is selected for the unstable habitat in most of the cases, while we find expansion events at most of the comparisons in the stable habitat. So it seems that both expectations are being met. There is stronger isolation and higher persistence in the stable habitat, and these results suggest that habitat persistence can indeed affect population isolation and population persistence, and it can potentially have an effect in the success of speciation as well. And this is where we are focusing right now, using phylogenomics and broader geographic sampling in order to see if we indeed get higher speciation in one of the two habitats. And with that, I want to finish my presentation and thank everyone that has helped for this work to be done, and of course all of you for being here today and the coordinators of the meeting that made our virtual presence possible. So thank you.
Thank you, Emmanuel. I think in the interest of time, we will move on to the next talk. So next up is Benjamin Wolfel. If you can share your screen.
Hello, yes.
Hi. There we go.
Hi, everybody. Welcome to my talk. I hope you can all hear me and everything is set up well. My name is Benjamin. I'm a PhD student at the University of Vienna, and I'm just about to finish up my studies and start my postdoc studies. My today's talk will be about a theory of oligogenic adaptation of a quantitative trait. So it will be at the interface between population genetics and quantitative genetics, and we'll talk about mathematical models. The motivation of this is to understand, based on the genetic architecture of a trait, meaning, for instance, the size on the genetic map, the allele effects distribution, the mutation rates, as well as the recombination map, what can we expect upon acting evolution will happen on an allele frequency level. So we want to predict, based on a certain genetic architecture, how the adaptive architecture will look like. The adaptive architecture will solely contain the recruited alleles, the ones that are driving phenotypic adaptation. An interesting question is, for instance, to discern the contributions of small versus big effect alleles. In our case, we focus on this adaptive architecture, and our description of it is via the joint allele frequency distribution of the underlying beneficial alleles. And we don't do this over time in generations, but over what we call phenotime, or just simply put as a function of the mean trait. On the top right, you see a classic representation of what we understand as allele frequency dynamics. However, typically these dynamics here of 4 different allele frequencies are studied across time. We don't do this in time, but instead as a function, as I mentioned, of the mean phenotype in a single population of a single quantitative trait. So let's describe our model in detail. We need, of course, several assumptions in order to create some predictions and contrast them later against simulations. And we hope later on also against empirical results, in particular from experimental evolution. Firstly, we assume that we have a randomly mating, so panmictic population of capital N sexual haploids. However, the model will also extend to codominant polyploids without any dominance. This means we can in principle display our population in such matrix form where we have individuals representing rows, and each individual carries a set of L slots that can be occupied by 2 different alleles. Accordingly, we can attribute a phenotype to each individual, and we can determine allele frequencies, and in principle L different allele frequencies, to describe the population state at any point in time. Accordingly, we will have L loci, and these loci are assumed to be biallelic. So we only allow for 2 alleles to exist, and these are under free linkage, meaning that recombination operates faster than selection. Furthermore, we assume that recurrent mutation can occur between these 2 groups of alleles. This means recombination acts fast, and mutation can act at any point in time in both directions between these 2 alleles. So we'll have one allele that we call small a at the I flocus, and this contributes no effect towards our quantitative trait. And then there will be a second allele, capital A, I here in black, and this one contributes a positive effect, gamma I, towards the quantitative trait. Of course, it seems a bit weird that we only allow for 2 types of alleles to exist, but one can simply imagine that these 2 groups could represent, for instance, the 4 different genetic bases. We may have on the left-hand side ATG providing no effect on the trait, while, for instance, C may provide a positive effect. Next, we assume the standard additive model for a quantitative trait, which means the allele effects gamma add up. So the trait value of a single individual is given by Z in green, and this is just given by a summation over the allele effects, using here an indicator variable eta I that is 1 if the capital AI allele is present and 0 otherwise. For example, we have such haploid individual at the bottom carrying 3 black alleles. The rest is white, so small a. This would then mean that this given individual has a phenotype Z given by gamma 2 plus gamma 3 plus gamma 5 for this 9 locus example. Later on, we also need a phenotype to fitness map. To that end, we use Gaussian stabilizing selection. This means that an individual with a given trait value Z maps into fitness WZ given by this bell curve, which is characterized one hand by the optimum trait value, Z opt. This is where fitness is maximal. And secondly, by the width, the measure of variance, which is the inverse of the selection strength. So the selection intensity being big means that this variance or this width is small. After all, this type of stabilizing selection will induce epistasis into our system, which makes the analysis more rich. Lastly, we want to track allele frequency or population genetic dynamics after an environmental change, meaning that this curve shifts to a higher value, so it shifts to the right, and it may also change its width. And then we want to track the population genetic allele frequency dynamics. And again, we do this not in time, but in phenotime, as we will see later. So we have some old trait optimum here on the left-hand side in yellow that is approximately at the mean trait of the population, such that at some time point 0, we will obtain a mutation selection drift balance, here called standing genetic variation, that is U-shaped, so not too much mutation. At some time point 0, the optimum shifts up to this blue set new opt, which is above the mean trait at all times, and is also higher than the ancient old optimum in yellow. Furthermore, this new optimum can wiggle around. We then want to track the allele frequency dynamics at all L sites in this blue phase before we reach the new optimum. Just to characterize some important dynamical variables, the mean trait is given by this quantity at the bottom, just allele frequency times the allele effect. And this allele frequency Pi is just the frequency of this black AI allele at the I locus. Mathematically, we do this by segmenting the entire dynamics into 2 phases. The first phase, where mutants are rare, so the mutant allele frequencies are low. In this phase, we can apply stochastic treatment because drift heavily dominates the dynamics, where we consider drift, so finite population size effects, mutation, and also selection. However, this selection is non-epistatic because frequencies are low. We do this mathematically employing a Yule branching process. Later on, once mutant allele frequencies are sufficiently high, so the underlying counts are high essentially, they move deterministically and we can apply classical differential equation approaches where we can now also correctly represent epistatic selection and the saturation once we reach the population size in terms of allele frequency count. Others have used similar approaches, such as in the previous work of my collaborators, or by Martin and Lambert for a single locus. Coming to the results, we can start by simply looking at the case where the allele effects across sites are equal and positive. Furthermore, we impose the rule that all the mutation rates across sites are the same. So the locus mutation rate at the I locus is just the same for any I. This is not necessary for our formalism, but makes the graphical analysis simpler here. And this work has been published and is available as a preprint on bioRxiv under this link and title. In the following slides, I will show you firstly the analytical predictions for the joint distribution of allele frequencies across the L sites. And secondly, I will contrast this against Wright-Fisher's computer simulations. The analytical predictions are shown in full lines, while the simulations are shown as density plots using disks. In our first scenario with equal allele effects, we will just stay with a simple system of just 3 loci. And we basically assume that at the time point 0, the optimum shifts, maybe the temperature drops to some catastrophic event, and then there will be some selection. We may imagine that, for instance, the quantitative trait could be body size, which now increases due to thermal improvements. Then we expect that the positively contributing allele regarding the allele, the trait will increase in number. And we now want to track exactly how this underlying vector of allele frequencies, this L-dimensional construct changes. And we do this not just in time, but we do this in the space of phenotime as a function of the mean trait Z bar. And to be precise, we want to view the population when the mean trait is exactly gamma. As a fundamental assumption, we need that the scale selection coefficient on any beneficial allele is larger than 1. This means that what we expect is as follows. So we see on the left-hand side 4 example realizations. So here from 1, 2, 3 up to 10,000, we will see essentially 10,000 replicates in our computer simulations. Now, if we view them, we get nothing else, but here we get an example of around 7 individuals and 3 sites. And if we observe at a trait of gamma, mean trait, then this should be the realization. If you count them, you'll always get NE times the positive allele. And we can just look at the sites marginally. If we would do this, this is represented on the left-hand side, then we would just get a single prediction for all the different 3 loci in terms of allele frequency density. This is clear because basically those sites are exchangeable as they have the same allele effect. However, we can also sort the sites such that the leading locus is represented in red always, and then the second highest allele frequency in blue, and then the third one as green. And then we'll see that even though they are exchangeable, mutation and drift ensures that there will always be a major locus. This is the locus contributing the most. And then we'll get such a split up of these densities in terms of order statistics. If we look at this system of 3 loci, we can now vary parameters and find out what's essential. And what we will find is that on the top, we consider a low mutation of a theta background of 0.01. Theta background is just, in this case, 2 times the locus mutation rate, because we have 3 loci. And we can increase this number clockwise and notice the characteristic change that occurs, which only depends on this theta background and is independent of the selection intensity. And you notice that comparing simulations as disks and predictions as lines, that these two fit very well. And secondly, that there's a characteristic change, which means that with higher mutation rates, the curves are drawn together. We can also consider different allele effects. This is represented in my doctoral thesis and had not yet been published in any paper. If we do this, then obviously individuals have different allele effects depending on the locus we focus at. Here I show an example of 9 sites. And then we have 3 different groups of allele effects. And we can again, of course, check this vector of allele frequencies and try to predict it. We now stay only with marginal allele frequencies because obviously now every locus may have a different allele effect. And by taking order statistics, we would get rid of essential information. If we consider just 2 loci, which is not essential, we can do it for more loci and also in initial intermediate optima, which is represented in our publication if you want to find out about it. However, for this presentation, I just do 2 loci, and I assume that the first locus has double the allele effect compared to the second. The red has double the allele effect compared to blue. And we again observe at different degrees of the mean trait, at 1/3 of gamma 1, 2/3 of gamma 1, and 1 gamma 1, where gamma 1 is the allele effect of the first locus. And we see again that simulations coincide with analytical predictions across different levels of the background mutation rate. Additionally, what we get when allele effects are different is that there is a dependency now on the absolute selection strength, which was not present before. So I conclude, for equal allele effects, we find that the background mutation rate is the sole determinant, and there is an independence of the selection strength, while for arbitrary allele effects, the background mutation rate still stays essential. However, additionally, we get a dependency on the absolute selection strength on top of that. Furthermore, the second formalism allows to treat relative pleiotropy. besides just different allele effects. Future directions in our research are to consider long-term dynamics, meaning the late, nearly neutral phase where we are reaching the new optimum, because so far we only considered the rapid adaptive phase where we have not yet reached the new optimum, then population structure, and furthermore linkage, starting for instance with asexuals. Lastly, I would like to say thank you to the Vienna Graduate School of Population Genetics that I have studied in, and to my collaborators and supervisor Joachim Hermison and also Isa Höllinger. Thank you very much.
Thank you, Benjamin. That was really cool. In interest of time, we'll move on. If someone wants to ask a question, you can ask in the chat or even by direct message to Benjamin. Our final speaker for this session is Chris Brower. If you'd like to share your screen. Are you there, Chris?
Yeah, sorry, I'm just. Okay, how is that?
It looks good, but we can see your next slide as well. Looks like.
Sorry about that. Okay, how is that? Good. All right, so thanks to everyone for tuning in. My name's Chris Brower. I'm a postdoc in the Molecular Ecology Lab at Flinders University in Australia. And I'm going to be talking about some of the work our lab has been doing with Australian rainbow fish. It's a bit of an evolutionary model for our group. We've been looking at how this group has evolved in response to the range of different climate regions that they inhabit. So in particular today, I'm going to be presenting a paper that has just come out in Nature Climate Change a couple of months ago, where we examine the potential role that hybridization might play in shaping climate resilience. And so the initial ideas for this paper were actually developed during some field work we did in 2017, which is when this photo was taken. We have on the left of screen Luciano Bejarrigorey, who's the head of our lab, Jonathan Sandoval Castillo, who's our bioinformatician, Peter Unmack, an ichthyologist who specializes in rainbow fish, Lou Benatchez, who's many people might know, he's a pretty well-known fish guy as well. Me in the background, Katie Gates, who was going to be speaking after me, but has been, I think, delayed in the field and didn't get back in time. And this guy here, Michael Hammer, who is another ichthyologist that works at the museum in the Northern Territory in Australia. And so as we get into it, I want you to keep in mind one sort of broad question related to conservation, and that is, what is the conservation value of hybrid populations? So historically, hybrids have been a controversial topic in conservation, and that's been due to a range of concerns, such as perhaps the genetic integrity of the parental species becoming diluted, demographic or genetic swamping, leading to extinction or reduction in fitness of the hybrid populations, and perhaps even outbreeding depression, as well as policy and legislative issues. Now, despite these potential challenges, I'm hoping to convince you that hybrid populations can be valuable for conservation. And so, just so we all are on the same page about what a rainbow fish is, we're going to have just a really quick look at rainbow fish evolution. They belong to the Athyrhinoformes, which are commonly known as the silversides. The family I'm talking about today is Melanataniidae, which are endemic to Australia and New Guinea. I've put here the evolutionary origin in New Guinea. So in the phylogeny, you can see here everything in the red box is endemic to New Guinea only, and then just down the bottom is the southern clade there, which has species in both New Guinea and Australia. But altogether, they're one of the most abundant and widespread families and the largest monophyletic group of freshwater fishes in the whole region, with 7 genera and probably more than 100 species, although some of them are really not well described at all. And so if we look at the distribution map, we can see several lineages in New Guinea, with just the southern group, which is indicated in the yellow and brown here, found in Australia. And then that's further divided into just the brown, the Australis group, which diverged from the rest of the southern group somewhere around 10 to 15 million years ago. And that's the clade that we're going to be talking about today. And one of the things that really stands out about the Australis clade is that they contain one of only 3 genera of rainbow fish that readily hybridize. And so when we're talking about the possibility that hybrid populations may be able to, it may help species to evolve to new environments. It's interesting to think about whether or not that was actually a feature that enabled that group to actually radiate and diversify across Australia, which is a huge, across a huge area in a whole range of different environments. And so looking at the genus Melanatania, we can see that they nowadays inhabit a whole range of eco-regions in Australia, from subtropical, tropical, temperate and desert. And their species distributions are really tightly constrained by the environment a lot of the time. And so we've done quite a bit of work on this, and these 3 figures come from a paper by bioinformaticians, Jonathan Sandoval Castillo, who published in PNAS a couple of years ago. And if we look at panel A here, now this is looking at 3 different species of rainbow fish, a desert, a temperate and a subtropical species. And when we build their environmental niche models, we see that there's minimal overlap between the potential areas that these species could inhabit. And then if we look at panel B, this is a plot showing what's called the warming tolerance quantity, and that approximates the amount of environmental warming an ectotherm can tolerate before performance drops to fatal levels. So this is really talking about sort of long-term acclimation. So rather than panel C, which is talking about CT max, or the upper thermal tolerance. And we can see here that the temperate, if we just ignore the y-axis here, this was from a transcriptomic paper and was looking at the number of differentially expressed genes. But if we just concentrate on the x-axis, we can see that the cooler temperate species has a lower upper thermal tolerance than the desert and then the subtropical, in that order. And also the more variable environment actually had a wider range of tolerance compared to the subtropical, which had the narrowest range of environmental variation as well. So altogether, that suggests that each species has evolved traits related to the climatic region that they inhabit. And so back to the current study, today we're looking at 5 different rainbow fish species found in the Australian wet tropics. We have 4 narrowly distributed habitat specialists, which consist of 3 cool adapted species. I've only got photos of 2 of them here. Melanitania aitchimensis, Malanda rainbow fish, and Tully rainbow fish, which are 2 species that are in the process of being described, but not formally described yet. And then we have Melanitania aitchimensis. Sorry. And so the 2 top species here occur in cool upland, montane, tropical rainforest streams. Aitchimensis is a warm lowland habitat specialist. And then we have the 5th species, which is Splendida. And they're a warm, really widespread, warm lowland generalist species. And if we look at the map in A, we can see that their distributions, within the study area span an elevational range of about 800 meters. And that each species, so if we look at aitchimensis and Malanda and the Tully rainbow fish, they occur in the higher elevation areas. Aitchimensis is way down in the lowland coastal flats, in a really restricted distribution. And lastly, Splendida occurs both lowland and in recent times has been moving up into the higher elevation streams as the climate warms. So this has only been happening in about sort of the last 20 to 30 years, and that raises some questions. And so Splendida are able to move further upstream and expand their range into the higher elevations as the climate warms. But what happens to the cool adapted upland species? Now they can't disperse because they're already in the headwaters, they can't move any higher, and they currently inhabit the only tropical montane rainforest on the continent anyway. What about acclimation via phenotypic plasticity? Now we know from previous work that rainbow fish that have evolved in narrow environmental ranges have a reduced plastic response to warming. So that leaves adaptation. Now, due to their narrow environmental range and their small population sizes, they have lower genetic diversity generally. And so that probably limits their ability to adapt in situ. And that brings us to hybridization. So the initial question, and back to the initial question of what is the conservation value of hybrid populations? And so that leads me on to my comic cover. I'm a bit of a comic geek. And the question are, hybrid populations the heroes we need to fight climate change? And so to answer this, we first constructed environmental niche models for each species to track range size variation throughout the Holocene into the future. We used genotype environment association analyses and genomic vulnerability estimates to try and understand how each species might be more or less vulnerable to climate change, and also whether the hybrid populations would be more or less vulnerable to climate change. And then we looked at the genome, the genomic regions that were introgressed, and tried to look for overlap with the GEA candidate genes. And we predicted that the narrow range species would lose more habitat hybrid populations would require less evolutionary change, and the introgressed genomic regions would be enriched for those climate-associated loci. So first, we identified 167 pure individuals using admixture. This is based on 344 individuals genotyped at about 14,000 dd-rad SNPs. And then we identified 85 hybrids that included Splendida, where the Splendida and whichever narrow endemic species both had an admixture Q value greater than 0.1 and all others less than 0.05. These classifications were well supported by the hybrid index analyses and new hybrid simulations, demonstrated high power of the data to resolve these classes. And our TrueMix tree identified that all the lineages were monophyletic, but the tree was improved by 10, including 10 migration events, 9 of which were from Splendida into the narrow endemics.
Chris, your time is about up if you want to wrap up real quickly.
Yeah, okay, I'm running behind. Okay, so the environmental niche models we used to try and predict the range loss throughout the Holocene, what their range size was and what was going to happen in the future. As predicted, the narrow endemics were predicted to lose up to 95% of their suitable habitat by 2070. The genomic vulnerability estimates, the top 2 panels show the allele frequency change required to keep pace with climate change. Panel C shows the population genomic vulnerability as a function of Splendida ancestry. And then panel D demonstrates the capacity for allele frequencies to change based on whether the adaptive allele is present. And so we see with the pure populations of each species, there's a greater number of the adaptive alleles not present in the population than for the hybrid populations. And just quickly with these statistics, we found only 4 of our 211 candidate adaptive SNPs occurred in introgressed regions. So this is perhaps not the most convincing evidence of adaptive introgression, but there is more to come. We're planning some whole genome work with these fish and sampling more fine scale across the hybrid zones.
All right, Chris, I think we have to stop there.
Yeah, okay.
Sorry.